so hard to open. Oh, I found it. Also, am I supposed to? Aren't you not supposed to get your flu shot if you're sick?
They say not to, but it's because. So the reason why they say that.
She came back and she's like, "Oh, you're sick." I was like, "Yeah, I just have a really bad cold lately." And she was like, "Oh, okay. So flu shot." And I was like.
Yeah, I think it depends. All right, so just a couple of quick notes before we get going today. The first one, maybe this is the only one. So Wednesday, review session, 4 to 6, I'll tape it. Plan on it being over in Sonrager 108. I'm just going to double check on the room and everything. I have to do that, but otherwise, that'll be the plan. 4 to 6 Wednesday. All right. So what I want to do to start is to give you guys about 90 seconds to look through the quiz from the weekend, and I want you to look through, and if there are particular questions that you're still not clear why the answer is what it is, write those down, and then we'll talk about them for a few minutes, or however long it takes. So go ahead and take a quick look there before we get going.
Yeah, there's 2 spots left, and they haven't been filled. All I need to do is submit the application. So all I did was like test the first one. So oh, well, it already went away. Like I literally got it 20 minutes ago, and it's already gone. I don't know. Let's have me that has all the problems has a good system.
Of course.
I also
figured I need to make a bigger for you guys.
severe anxiety about shots, and I don't know why. Actually, I do know why, but I don't know why. They don't though. That's the thing. I always like sit there and I'm like, wait, are you done? And they're like, yeah. And I'm like, wait, what? And I don't even because I had staph infection once in my knees. In my knee, and then spread up my leg, and then it took me to my heart, like something about killing me or something. I don't really know. I didn't understand that as a fifth grader. Yeah, I fell on the playground and I got staph or something. I still have a scar on my knee. The last thing I've done on my computer.
All right. Any questions on any of these? So where are there still questions? Haley. Okay, so let's go to that real quick. This one here, which statement is correct? Is that the one? Okay. So which statement is correct concerning C. diff? In some of these, the answers are actually part of what we'll talk about today. So like the first one up there, it's a major cause of pseudomembranous colitis. That's something we'll talk about like really briefly today. It's kind of a hallmark characteristic of C. diff infections. It is an anaerobic. So it is an organism that is, we oftentimes consider it a strict aerobe, though I think it can tolerate just a bit of oxygen. It is a spore former, and that is common among all the Clostridium family members. Tetani, botulinum, they all are spore formers. And then this is actually, let's see, can be treated by stopping antibiotic treatment, fluid replacement, and if necessary, with antibiotics such as vancomycin. These are all kind of ways that it can be treated. So this wasn't necessarily in the what I had talked about in the slides, but these are ones that I had added on to the end of what was in the mini lecture, and that's kind of where we're going to go with the conversation today. So this wasn't explicitly in what you guys had read, but those are all true. Okay? Do you have other questions about it before I? The one that says all of the above, which is really only pointing to this one right now, but I should probably explain like all of the answers are correct. So it would be the all of the above one. Danica. Yeah, so which of the following are found primarily in the intestines? And there's probably a couple of ways I could have redefined this question. Thinking about it after the fact. One of them could have been to define it as which is found found primarily in, and I could have stated which section of the intestine if I'm talking large and small. I think in the way that I had written this one, I was intending to mean large intestine, because if I'm not mistaken, the answer was gram-negative bacilli. And what we find there primarily are Bacteroides, which are aerotolerant gram-negative bacilli, and then we have the Enterobacteriaceae, which are a larger, it's more of a class rather than a specific genus of those gram-negative bacilli cells. And those include like your E. coli, Klebsiella, so some pathogens and some non-pathogens. If we were talking small intestine, how might the answer differ? Possibly gram-positive cocci in the enterococci. You might also argue gram-positive bacillus to an extent with lactobacilli. We oftentimes think are found within the small intestine as well. Those would probably be much better answers if we were discussing the small intestine. So in part, that's my fault just for not defining that question really well. Any other questions? Zach, did you have one? No? Okay.
What was the answer for number 10?
That one? Which of the following is an anaerobic gram-negative rod? This is the Bacteroides. So the Peptostreptococcus, those are gram-positive. Streptococci bacteria typically are. Lactobacillus are gram-positive. We'll talk about these more on Wednesday. Shigella is an example of an Enterobacteriaceae. So this would also be a gram-negative rod, but it's a facultative anaerobe, which is true for all the Enterobacteriaceae. And then Enterococcus is another gram-positive coccibacterium. Okay. Yeah, and so again, this could have probably been, if I had better defined this as like aerotolerant anaerobe or obligate anaerobe, the answer probably would have been more clear. Absolutely. Danica? Yeah, it's a great question. In the past, what I've done is I've told people you should be able to, like, let's say I tell you, okay, which of these bacteria are found within the gastrointestinal tract, and I might give you a list of like 3 or 4 of them, and based on the characteristics that you know about that microorganism, you should be able to identify which one would be in the gastrointestinal tract. I thought about it a little bit differently this time. What I more want you to be able to do is know the characteristics of the microbe, at least, and when I say know the characteristics, know the ones that I've put on the slide. Okay? That's what I'd like you to have for them. That information may be helpful when it comes to the exam. Okay? And the reason why I'm not focusing so much on the specific type of bacteria here is because our understanding of the human microbiome is honestly changing I would say it's changing every single year. Like what we think is there right now, largely, we focus primarily on aerotolerant anaerobes and facultative anaerobes when it comes to the gastrointestinal tract, because that's what we can grow real easily. It's really challenging to grow an obligate anaerobe. And so in the past, our identity of them has been pretty weak, has been limited by just how we culture bacteria as a way to identify them. Sequencing DNA has allowed us to get a better understanding, but sequencing doesn't necessarily tell us if they're alive or dead. So every year, the information that we get changes just a little bit. So what I tell you now may be different in like 15 years from now, essentially. Other questions? Okay. So I wanted to move into the lecture, and I'm going to skip a lot what I talked about. But today, what we're largely going to focus on here is we're going to talk a little bit about how the environment can influence the microbes. So based on what's there, what's not there, what the environment is like, how does that select for different type of microbes? We'll talk about commensal organisms, particularly those in the GI tract, and then we're going to go back to the conversation of antibiotics, okay? But before I jump to all of that, I want to go here and I want to just kind of give a couple of comments on things that I brought up real briefly in the lecture and want to expand on now. So one of them are these short-chain fatty acids. And when we say short-chain fatty acids, we're talking about products of fermentation: lactic acid, butyric acid, propionic acid, acetic acid. And these are going to be products of microorganism the fermentation by microorganisms. We think, or at least some of the newest findings in studying the human microbiome suggest that these short-chain fatty acids that are produced by bacteria actually contribute to human health and also contribute to cell physiology. So for example, it's thought that some of these actually have a role in controlling the growth or regulating the growth of cells that make up the colon or that line the colon. And there's some importance here. By regulating cell growth, it's argued that these short-chain fatty acids actually can be involved in regulating uncontrolled growth or colon cancer, or early initiating events of colon cancer at least. Another one where these short-chain fatty acids are thought to be really beneficial is that they can actually help in maintaining the tight junctions that hold together epithelial cells that line the colon. And in helping to hold those cells together, it reduces their permeability and reduces the chance that you're going to find microbes from the colon exiting the colon and making it into the surrounding tissue. Benefit of that, lower inflammation. Because as soon as those organisms make it outside of the colon, what we find is that there are resident innate immune cells, macrophages for example, which are ultimately going to contribute to preventing infection in areas of the body where we don't want bacteria to be, or where our body isn't designed for bacteria to be located. So by maintaining these tight junctions here, this can actually reduce inflammation. One argument is that some of the diseases that we contribute or attribute to constant gastrointestinal stress, like irritable bowel syndrome, in some individuals, Crohn's disease. So those diseases that we often think might be in part immunity or disruption of the microbiome. There are some researchers that believe, based on the findings that they've seen so far, that could be explained by the production of these short-chain fatty acids. And again, the types that are going to be produced are going to be dependent on the microbes that we find within the GI. There's some good data to suggest that short-chain fatty acids have a role in a cell's response or its sensitivity to insulin as well. Finally, there is some evidence that bacteria actually do produce neurotransmitter-like molecules like serotonin, dopamine. So it has been hypothesized, though by no means proven or supported without a reasonable doubt, that microbes in some way contribute to communication between what's happening in the gastrointestinal tract and in the brain as well. All right. So let's take a look then at superinfections. I've put a definition up here, and I'm not going to ask you guys to read or to per se just repeat the definition, but what I want you to do is read it, put it into your own terms, or give an example. I'm going to give you about a minute to work with somebody else if you'd like to, or alone, just to take this definition, a secondary infection from a different microbe, often resistant to the treatment used for the first. Give an example or redefine that in your own words. Okay, go ahead. So the resistance might not have necessarily to do with whether it's broad or narrow, but whether the organism has the mechanism for resistance. So like if a mutation has occurred. But broad and narrow do play a role potentially in this as well. All right, so what is either an example or a different way to define a superinfection? Go ahead, Zach.
I totally guessed, but maybe a mutation of the first infection agent could become resistant to that antibiotic and then superinfect.
So you're saying one where maybe like you take an antibiotic for a couple of days, the infection goes away, or it appears to go away, and then it comes back later. Some people do group that with superinfections. Yeah, absolutely. Other examples? Sid?
I thought that would be like if Kaylee got infected or something. She took an antibiotic, the bacteria became resistant, and then like went to Zach, and then Zach took an antibiotic and it resistant, and then it changed, and then like went to me or something. Just hopped around.
Okay, so that's somewhat similar in the same way, right? That it's one organism that, upon exposure to the antibiotic, there's some sort of mutation that selects for a subset and allows for it now to the population of it to be resistant. Yeah, and I think that kind of does fit as well. Other examples? Okay.
One that like piggybacks on another.
Yeah, piggybacking isn't uncommon. So ones that we do actually see, and some, I mean, this one is kind of saying that resistance to treatment, which kind of implies the idea of antibiotics. We see sometimes superinfections occurring in the absence of antibiotics as well. So for example, one that we do find is actually influenza, will infect the lungs, cause damage to the cells in the lungs, and that makes the inflammation and the damage that's done actually make the lungs a great site for secondary infections. What kind of secondary infections often occur after flu? Pneumonia is the biggest one. So that could be an example of a superinfection. Okay? Another one that we're going to talk about today, C. diff, is a great example of a superinfection. Okay? So where I want to kind of preface this to begin is just to say that the microorganisms that are inside your gastrointestinal tract, and that's where we're going to focus on today, you can kind of think of as your gut is a site of one, evolution, and then two, constant warfare between microorganisms. Okay? In the sense that we've got some microorganisms that they've got different ways to either attack or defend against invading microbes. So for example, we have some that have the secretion systems and what are known as bacteriocins, which are basically antimicrobial proteins. So Pseudomonas, Vibrio, Bacteroides all produce these antimicrobial peptides or proteins that are secreted from the cell and will inhibit the growth of other species of microorganisms. Okay, so that's kind of an attack mechanism. Others just outcompete for food. So we've got E. coli, Salmonella, Clostridium are all going to some organisms are going to compete for food like sialic acid and fucose. which are going to outcompete E. coli and some of these others. We have some inhibitory mechanisms within us, bile salts. Short-chain fatty acids produced by some microorganisms can be inhibitory to others. Lactic acid is a great example of an inhibitory mechanism, and we'll come back to that on Wednesday. So there are ways in which microorganisms contribute to that environment inside the gastrointestinal tract. There's another one that's not listed on here, but we should make sure to make note of because we'll come back to it today. And that's this one called colonization inhibition. And maybe a metaphor for colonization inhibition would be, let's assume that we had enough people to fill up all the seats in this room, and then somebody else were to come into the room, there are no seats available, they would not be able to quote-unquote colonize or to sit down in an available seat, okay? Or you might think of like in an apartment complex, there's a certain number of units that can be rented. Once it's filled, nobody else can enter or can become a resident of that apartment complex. So the inhibition is that our gastrointestinal tract, particularly the large colon, or the large intestine, is not only covered in mucus, but also is coated on the surface with different type of bacteria that have actually attached to the surface of those cells. By attaching, prevents potentially pathogenic organisms from attaching as well. Okay. So keep that in mind as we start to talk about C. diff. Questions on those concepts? All right, the big one out of here is the colonization inhibition. We've talked about broad and narrow spectrum antibiotics. And one of the things that we've said about the difference between these two, broad spectrum will target a large selection of happy microbes, narrow spectrum, one subset. Okay? I want you to think about this then in terms of the gastrointestinal tract for a minute. And one of the things that we have to consider here is assume somebody's got an infection. And oftentimes these days, unless the infections are ones within the bloodstream, oftentimes these drugs are taken orally. What effect is a broad spectrum antibiotic going to have on the inhabitants of the gastrointestinal tract? I'll give everybody a minute to think about this one before you answer. So I want you to just jot down an idea for yourself. What's the effect going to be on the resident microbes within the gastrointestinal tract if you're taking a broad spectrum antibiotic? All right, Jaden, you had your hand up first. What would you say?
It's going to kill all the bacteria, including the good ones.
Yeah, does anybody want to add to that before I continue, Danica?
That's why you would take like a probiotic with it.
Ah, yeah, so who's heard of the idea of taking probiotics alongside an antibiotic? We're going to come back to that in a minute. I have some thoughts on it, or at least I'll give an opinion maybe. Yeah, so, I mean, antibiotics aren't distinguishers in terms of who's good and who's bad, right? Antibiotics distinguish based on, does an organism have this particular protein that's going to be targeted or particular pathway that's going to be targeted or not? Some of the best antibiotics or some of the really good antibiotics, I guess, would be ones that we consider to be narrow spectrum. For example, isoniazid and ethambutol, you may remember, targets mycolic acid synthesis, targets the mycobacterium exclusively. They're the only ones that produce it. And that's a really good thing, because taking that antibiotic then isn't going to affect or disrupt the rest of the bacteria in the gastrointestinal tract. And we have to think too about all the bacteria that actually would come into contact with that antibiotic. It's not just as it goes down the throat to the stomach, but through the gastrointestinal tract, through the small intestine, large intestine, and then where? How do excess antibiotics get disposed of from our body? Waste in terms of what? 3 routes. Defecation, that's one. Urination. Vomiting, guys, vomiting. So most likely though, most of them would be through defecation or urination. And we'll talk about the effects of that on Wednesday, particularly in the female urogenital tract and the downsides of antibiotics to the health of the female urogenital tract. Are there benefits, though, of broad-spectrum antibiotics? Go ahead, Zach.
If you have super infection,
Maybe.
if you have one or more, or you have 2 or more bacteria that are infecting you, if you have a broad spectrum,
Okay.
let's say one is gram-positive, one is gram-negative, you can take them out both, 2 birds, 1 stone.
Okay, not a bad idea.
Game over.
Not a bad idea. Jaden?
Maybe if your infection is so bad that bacteria is taking over everywhere.
Okay. C. diff would be an example. We'll come back to that one. Danica?
I had a question, like, are there diseases where you would have, like, multiple bacteria growing? Like, do they know that there's
That's a good question. I can't think of any. I can't think of any necessarily off the top of my head. But I mean, I think Zach's point is well taken, though. That like, if you wanted to stop a super infection, you might treat with, or if you
Live through the stomach.
yeah.
Would that be like something that you could put on a capsule of a probiotic to like have it be extended release?
So you're talking like a bicarbonate or something like that, or even a capsule that doesn't break down during the 8 hours exposed to the stomach, but might break down. And that's a really good idea. That's actually an excellent idea. So what you're thinking about is provide it some way that it survives that condition and then that's a great idea. And I would bet that my guess is somebody's working on it, but I don't know for certain. The other way that some people, what's another natural way? Some bacteria have the ability to form what? Spores or endospores. What's the benefit of an endospore? Highly resistant to environmental conditions. You could argue that if you want to replenish, now lactobacillus aren't ones that form endospores, but there are some bacillus species that are often found in the gastrointestinal tract. What if you could deliver a spore? Survives the acidic conditions of the stomach and then flourishes once it gets into the small intestine. That actually has been proposed to treat some infections, actually. So. All right, I always look at it this way. You're gonna perform an appendectomy on somebody. Which of these two are you going to use to cut that person to take out the appendix? Okay? Are you just going to go for blunt force? Or are you going to go for a more tailored approach? Okay? And this is where the conversation is right now, is which of these is the better option. And neither of them is really wrong. It's just maybe which is the better given the context of the situation. Right? Yeah, right. They both get the job done, but one might just require more recovery time than the other. Maybe more prone to secondary infections. All right? Yeah, exactly. So let's just look at what we've kind of talked about in a slightly different light. So this would be a normal microbiome, all these different colors representing a diversity of microbes. And then treat with an antibiotic. What's left behind are those that are resistant to the antibiotic. And then provide enough time. I mean, and now the room is free. Imagine like half of us leave the room, the seats are open, and now we get some resistant bugger or some malfeasant individuals coming into the room and taking over our space. Okay? So that's what we're talking about here. And the question becomes, how do you stop this? In the context of, I'm going to skip, or I want to give one example of where we see this real quick. And this one I don't want you to necessarily memorize. I'm putting this out there as an example of where we see this in the context of clinics. So this is just a graph showing a number of different antibiotics. The ones on the top here are targeted specifically not on gram status, but rather which ones use oxygen. So these are against organisms that are primarily anaerobic. These mechanisms down here are ones that have minimal anaerobic activity or more aerobic microorganisms. And then this is looking at the change of vancomycin-resistant enterococci, so a gram-positive coccibacterium found in the intestines, in the presence of these anti-anaerobic antibiotics or these anti-aerobic antibiotics. And what they're trying to show you is that when you target particularly organisms that perform an anaerobic form of respiration, this clears out the gastrointestinal tract because most of the organisms there are anaerobic, and it creates space for population of vancomycin-resistant enterococci to increase. Okay, so this again, just I bring this out because this is a problem, and it's not just related to C. diff. We find this with other types of infections as well. So from the perspective of a clinician or a pharmacist, what you've got to figure out is, okay, so we have to look beyond just the drug interactions now. We have to actually start looking at what's the microbial interactions that might be occurring as a response of how we treat these infections. I just want to give a couple of background slides on C. diff. Okay, and some of this you may know. Some of you may have personal experience with this. We'll come back to that in a minute. Gram-positive bacillus spore former, opportunistic infection. And if you'll recall, what that means is it itself isn't it doesn't cause disease without there being some weakening to the host in some way. In the context of C. diff, what this really means is that there's freed up space. We find a loss of colonization inhibition because broad-spectrum antibiotics will wipe out the natural microbiome and create now free space for Clostridium difficile to colonize. Some of us are asymptomatic carriers. For many of us, C. diff will come into the system and then just exit within 24 hours. Because it can't attach on to any of the epithelium that line the GI tract. And a common treatment for it is vancomycin, which is a broad-spectrum, very harsh drug, not the best of therapeutic indices. Okay, so not a first-choice drug by any means. We actually have seen an increase over the past 15 years or so. There's been a gradual, pretty linear increase in the number of cases where somebody who is discharged from the hospital ends up getting a C. diff infection after leaving the hospital. This can occur because of a number of reasons, but in most cases, these people or these individuals, patients, have been put on antibiotics for some reason. Maybe they came in with an infection. Maybe they were put on antibiotics prophylactically because they came in for a surgery. And so to prevent widespread infection or sepsis to occur after that surgery, to keep that wound site clean, maybe they were put on a broad-spectrum antibiotic for a short period of time. And what we've seen is that the amount of cases is actually about tripled, or has been predicted to have tripled from 2001 through at least 2014. Okay. 500,000 infections in 2011, 29,000 deaths. The death rate isn't that high, but there's a lot of cost. You have to think about 500,000 infections. And has anybody had experience working with individuals who have C. diff? Good number. Okay, so what practices are you expected to PPE, meaning what in terms of C. diff? Well, yes, that's what the acronym means, but what does PPE look like for C. diff infection? Gown, mask, gloves. What my friend and I used to call in graduate school, basically the human body condom. Okay, so it's a protective barrier, right? It is gloves. It is mask, goggles. No? Okay, I wasn't sure. Usually a gown, and then when you exit the room of somebody who is infected with C. diff, what are you expected to do? What is the exit strategy? Good. There's a specific garbage can for everything, right? Do you wash your hands before or after taking all of that off? Yeah, because there's a possibility you may contaminate your hands if you wash your hands beforehand. So what's the purpose of that then? Why take the energy to put on the personal protective equipment and then also to strip it all off and throw it in a waste can before you leave? Why do we do that? Why do you do that, Paige?
Well, I feel like in most of the cases that we've seen it at least, like, we're in a place where there's elderly people, so the spores can travel from one place to the next, and even though you might not, like, be infected by it because we don't have, like, poor immune systems or aren't on antibiotics, it can travel from room to room and
Yeah.
someone else.
Yeah, I think that's a really important point. So, like, yes, there's some protection to yourself, but I think in the context of the settings where we find C. diff infections most often, it's really to prevent spread to the other patients, folks who are immunocompromised or who may be on broad spectrum antibiotics already. I think that's excellently put. Absolutely. All right. Symptoms. And then I do want to point out that about 25% to 40% of the infections may not occur until like 10 weeks after. So somebody may go home, and the reason why it may take so long is because for any infection to occur, we have what we call an infectious dose. There's a certain amount there's a certain amount of the bacteria, a certain number, colony-forming units, that need to actually build up before you experience the signs and the symptoms of the illness. So for some organisms, I'll use E. coli again, it's like 1 to 10 cells of pathogenic E. coli is enough to cause disease. Listeria, it's like 10 million to 100 million. I'm not sure what the number is for C. diff, but this could explain why there's this period of time between once you may be first exposed to it to the time when you start to experience some of the symptoms of the illness. We call that period of time incubation time, and that's something we'll come back to at the end of the semester. Want to point out just a couple of the features that we see here. Okay, so basically it disrupts the lining of the colon, and it leads to inflammation of the colon, and usually presents in this pseudomembranous colitis. So we get these little papules or pustules, if you want to call them that, where we've got damage to the tissue. This is caused by a toxin that C. diff is producing. Okay, so this is where we create that damage. It leads to inflammation, increases the likelihood of further illness or even sepsis potentially, as this weakens the membrane barrier of the colon as well. I'm gonna skip this slide here. This is kind of just like what are the additional costs, and here we're talking more about like financial in some cases. So I'm just gonna kind of skip that for right now. But one of the things that folks have started thinking about is, okay, so how do we actually clear these infections? As I mentioned, vancomycin is a common methodology right now. Vancomycin, a good last resort drug, but we do find that there are vancomycin resistant strains of Clostridium, and we already talked about vancomycin resistant enterococci. If that's resident in the gut as well, treating with vancomycin could lead to a secondary superinfection with VRE. So the idea is what? What is an alternative idea? Yay! Okay? Eat, poop, and live. Instead of eat, shit, and die, eat, poop, and live. Okay? This is actually being used. So, let's talk about this for a couple of minutes.
Do they just directly?
Yeah, so there's 3 potential methods. There's 3 methods by which this is done. And the first time I heard about this, it was about, so the first time I heard about this was about 7 years ago, and the only image I had in my head while listening to a talk was Austin Powers, where like he's trying to take a cup of coffee and it's actually Fat Bastard's stool. So anyway, tastes a bit nutty. All right.
And now I know why.
So how is this delivered? 3 potential mechanisms right now. I think the most common, if I'm not mistaken, is actually through the nose. So they'll take a tube, they'll put it through the nose, through the stomach, and they'll just feed you poop that way. Okay? Feeding people poop. Second way, rectally, which I think is less common, but is another option. The third way is freeze-dried pills. So, in that one, it would be, we talked about spores. They choose certain types of sporulating bacteria that they would put into basically a freeze-dried pill, and you would take that orally.
I would do the pill.
Yeah,
Is that
okay.
really, like, have they found benefits to it?
To that particular mechanism? I don't know if there are benefits, but if you think about the cost, so I mean, think about the cost of it. A freeze-dried pill is something you can do at home. I don't know how to, and I know how to swallow a pill, but I do not know how to take a tube, force it up to my nose and through my stomach.
You don't know how to do
No, of course not. All right? Yeah, exactly. I'm not that kind of doctor, just a reminder. Okay, so I think that there is, now I don't know how well it works, but that is something that has been proposed, and I'm not really sure where they are in the studies with it. So let's take a look at this real quick. I just want to take a look at the data here to kind of show you what the potential benefits are. And this is becoming more common. I also want to point out that your poop donor, okay, does not have to be like a blood donor, where you have to match for AB blood typing and whether they're AB positive or AB negative. All you have to find is poop, and usually you want to match with somebody who has a similar diet as you. So like if you are a vegetarian, you want to find another vegetarian that can donate poop, okay, to restore your natural microbiome. Oftentimes, the best person to donate is a family member you're living with. That's the best. That's the best, okay? So here is the data. The y-axis here is percentage that are cured without relapse. So they provide the treatment. There's no relapse of a C. diff infection. And this was first infusion of donor feces. Overall, how many, if they had to do fecal transplant multiple times, what was the overall cure rate? And then with vancomycin or vancomycin with a bowel lavage, which is basically cleaning out your colon. Okay, so rinsing it out with a wonderful saline solution or something like that. And if we look at the data here, I mean, fecal transplants, 81%. In other words, out of the 16 people that were a part of this study, I guess that would put it at 14? 14 of them, probably. 13.8. 14 of them on the first time were cured with a fecal transplant. It might have been 13, somewhere in there. I don't know. Vancomycin, on the other hand, 30% experienced relapse. Okay? And if we think about it, there's a couple of potential reasons. What is vancomycin's broad spectrum? So what does that mean in the context of using that to treat a C. diff infection? What's that?
Isn't it clearing everything else and keeping it around?
it's clearing everything else that might repopulate the gastrointestinal tract as well. So not only is it keeping at bay C. diff, which it's designed to do, but again, there's collateral damage here. There's an effect on the other microbes. So this came from a paper last year. So rather than carpet bombing germs into submission with broad spectrum antibiotics, we will need more laser-like approaches to develop drugs against specific pathogens, minimize damage to essential symbiotic microbe species, and preserve community structure and function in a healthy microbiome. In other words, we need drugs that are even more narrow spectrum than we have right now. And so there are some that argue if we want good antibiotics for the future, we need tailored drugs. Ones that could target one gene that is unique to C. diff but doesn't target the remaining microbes that are found within the gastrointestinal tract. That would be an ideal situation, right? We'll stop there. So for Wednesday, the homework assignment is another quiz. Again, take it as many times as you want. And I'll be updating, if you got an 1.8 or above, I'll be updating those scores later today. All right, have a good afternoon, everyone. Enjoy the Packers game tonight.